Akkor én tudok fölvenni?
Igen, indítsd el a felvételt, én is elindítottam.
Okay, before we start recording, I just want to emphasize that if you don't want to be shown, then please turn off your camera. We'll record the lecture and then we'll post it on our website. This is already the start of the summer. This is an unusual event. We have institute seminars throughout the summer, or at least throughout June. And it's even more exceptional because we have talks outside of mathematics. And our today's speaker is Dénes Ádám from the Institute of Experimental Medicine, who got his PhD from SOTE in 2007 after completing his undergraduate studies at ELTE as a biologist. And he spent 6 years in the United Kingdom in Manchester. And since 2013, he is working at the Institute of Experimental Medicine. He got a Lendulette grant in 2016 and in 2017 an ERC grant. So the floor, the imaginary floor is all yours, please. Kapcsold be a mikrofont.
Can you hear me?
Yes.
Yes.
Great.
Perfect.
So it is great pleasure to be here. Thank you very much for the opportunity to present some of the data and some of my thoughts to you. Although I was asked to talk about coronaviruses and specifically the coronavirus infection in the brain, I think there is not enough data at present to give a full lecture on this. But what I would do instead, I would tell you a few things about the brain and how inflammatory processes manifest in the brain and why they are important, as these provide a lot of clues to understand the potential impact of coronavirus infection better on the brain and on the whole body. And you will see where we come from, because basically this is quite a bit based on my previous research and those kind of stories we have been working on with multiple labs in collaboration throughout Europe. So what I will talk about is in front of you. First, I would like to briefly elaborate upon why inflammatory processes are very special in the nervous system, and touch upon a little bit the specificities of the brain microenvironment. Then we will specifically talk about how the brain's immune system can deal with viral infection. Then I will show you a few examples on how inflammatory processes, either central, which take place in the brain, or systemic, which takes place outside the brain but impact on the brain, would change the processes our brain performs in health and in disease. And then I would try to use this information to guide you how this knowledge might help us understanding what is going on in the brain in COVID-19. If you are interested in the mechanisms a little bit more, we have recently published an opinion piece, which is here available since 2 days. And this would touch upon a little bit the major topics I will talk about today. So first of all, the brain is an extremely complex organ. Probably all of you know that, but I would like to point out two things actually concerning this. First is the neurons to function in the brain needs an extremely well-regulated microenvironment. And this is why the brain has its internal milieu, and it has to be actively controlled for to maintain this state. And a second thing is a huge energy demand of the brain, which only gives 2% of the body weight, but would consume 20–25% of the total oxygen and glucose, which would circulate in the body. So the brain is really fine-tuned to take this energy constantly to perform its actions and to regulate all the peripheral organs. If it doesn't work, this will lead to a number of diseases, and you will see how inflammatory processes can impact on this.
Can I interrupt you for a second?
Yeah.
There is a little yellow rectangle in the middle which says, please move this window away. Can you move that away?
Yeah, I try.
If it works, fine. If it doesn't, it's okay. It's not.
It doesn't go anywhere. I can try to reshare the application, or.
Yeah, I think just unshare it and share it again, and let's see how it works.
Okay, just a second. Is it better?
Yeah, the usual Windows trick. Just
oh,
do
fantastic.
it.
So in order to maintain this huge circulatory demand, but at the same time to keep the brain microenvironment isolated enough to allow neurons to function, there is a very special circulatory system in the brain. And you can see here that most of the brain arteries go outside the cortex, the surface of the brain. And if you look at this image, they are kind of located in between the brain meninges. And then they dig deep into the brain parenchyma, but very, very special cells follow their route inside the brain and isolate this kind of vessels from the brain microenvironment. If you look at this image, this is kind of a jungle of processes of different glial cells, which are kind of supporting elements of the brain. And these processes end on these blood vessels. And if you look at the cross section, what is very special about the brain that we have endothelial cells which form the wall of the blood vessels, and these endothelial cells are very tightly linked to each other, so that whatever is circulating in them, which is in this case blood, certainly a lot of mediators and cells, cannot passively get through these tight junctions. And around this, there is this really, really tight layer of these end feet of astrocytes and also other supporting cells, making sure that whatever comes out is properly controlled. And a lot of things, for example, immune cells are not allowed to run into the brain parenchyma, as it is seen very widely in peripheral organs. So if you look at the inflammatory processes in the brain and why they are so special compared to peripheral organs, we can mention a few points. For example, there is quite open circulation in a way. It's not open in a form of insect circulation where there is a hemolymph. It is closed, it's circulating inside blood vessels, but whatever is in the blood vessels in the blood can get to the peripheral organs. For example, if a blood vessel enters the spleen and gives rise to capillaries, there is quite a high level of diffusion from the capillary, and also a lot of substances can get through the wall of the blood vessel to the parenchyma, whereas in the brain we have the blood-brain barrier, which is composed of partially these tightly linked endothelial cells and also this perivascular glial membrane, which limits the diffusion of substances, even limits the diffusion of glucose and many things into the brain, to make sure that the microenvironment is properly regulated. In the peripheral tissues also, we have diverse population of specialized immune cells which can run into tissues and perform their surveillance activity. In the brain, it's not allowed. I will show you why. And therefore, we have special cells called microglia, which provide the first line of defense in the brain, kind of the main immune cells of the brain. And what is very important, we have very high level of regeneration in most tissues. If you cut your finger, the structure will be close to normal in 2 weeks' time, whereas in the brain we have very limited capacity for injured neurons to regenerate. And this is why we have to protect them, because if there is inflammation in the brain, if we just have a cut in our finger, this could this could make 2 centimeter long tissue inflamed. This length of tissue in the brain, if it gets injured in the motor cortex, you will never be able to move your hand or your leg. So this is why it is very important to keep this tight and regulated. And there are some parts in the brain which are linked to the brain ventricles. These are so-called circumventricular organs, where these barrier structures are not that strong. And I will come back to this because these structures are the areas for intense neuroimmune communication, and these are the structures where, for example, peripheral inflammatory mediators can reach the brain and change the functioning of the neurons, and also change our behavior or mood or taste or appetite, or cause fever, for example, through these sites. Now, why is it so important to keep these immune cells out? What I'm showing you, you might recall on the upper image, this is an image of a blood vessel in the brain, but it's an injured brain. It is after a stroke. And you can see that this very nice and tight structure, the blood-brain barrier, and the surrounding glial structure is completely disintegrated. And when it happens, you have a lot of plasma proteins from the blood, and also immune cells which can get into the brain parenchyma, and it causes inflammation there, but it also changes the way the brain functions. And just one example, you can see here granulocytes which produce enzymes that can digest the wall of the vessels in the brain. You can see them also here coming out of these injured blood vessels. And if this happens, they would produce toxic mediators which would kill neurons. This is just an example. This is a cell culture. This big thing is a neuron, and you can see these small white blobs. These are neutrophil granulocytes, inflammatory cells, which can get into the brain when the brain is injured. And when it happens, these cells get activated, and they can pump out different proteases, enzymes, and these little strand-like things from the cell, and they can get degranulated. And if it happens in the proximity of neurons, you can see that within minutes, the membrane of the neuron breaks completely and the neuron dies. So it's very, very important that even if there is injury in the brain, to provide some sort of regulation for inflammatory processes. Otherwise, neurons will die very quickly and the brain will no longer be able to function. Now, how the brain still can maintain its integrity and can defend itself against inflammatory immune challenges is due to a very special population of cells which we call microglia. And these are kind of macrophage-like cells, but a very special subset of macrophages. And they have been seen since the beginning of the 20th century. And also identified by really famous people like Alzheimer, that in the diseased brain, these cells can change their conformation. They become amoeboid or phagocytic. And this process at the same time appears to contribute to degeneration of neurons. So this knowledge is quite old, but the mechanisms, how it happens and how we could interfere with this is still very not understood. Now, I just want to quote a lawyer and philosopher because it's very interesting statement considering the importance of activity and also the importance of proper level of control. So it is the common fate of the indolent to see their rights become prey to the active. The conditions upon which God has given liberty to man is eternal vigilance, which condition if he break, servitude is at once the consequence of his crime and the punishment of his guilt. So microglial cells are very not indolent because, as you can see, this is an anesthetized mouse. And in the mouse brain, these microglial cells, which are red here, on the left side, you constantly survey the microenvironment with their little motor processes, and you can see the blood vessels in green, and you can see that every single blood vessel is surrounded with these little motor microglial processes. If you look at the video on the right-hand side, now you can see that these motor microglial processes interact with neurons and constantly check the condition of neurons, and they do this with almost any kind of cells in the brain to kind of monitor their status. These are the cells which would phagocyte injured neurons when they are dead, And these are the cells I will show you which contribute to defense against, for example, viral infection. At the same time, these are the cells which will produce inflammatory mediators in the brain. So their proper functioning is extremely important. And if we look at the three-dimensional reconstruction of a brain vessel here, you remember on the top of the cortex, this vessel digs deep into the brain parenchyma. And you can see those small red microglial cells located everywhere around these blood vessels and in the tissue. So this is a very, very important immune barrier to notice any kind of disturbance in the brain and to very rapidly respond to this. How rapidly can microglial cells respond to infection or injury? This is a very interesting paper, one of the first ones which could show really nicely how actually the different types of cells, including microglial cells, can function, and try to show you the video. So this is the red thing is a pipette inserted into the brain, and ATP, a danger molecule, which tells the tissue that there is injury or cell death, is injected, and you can follow how quickly these microglial processes within minutes surround and isolate this area. If any kind of injury happens, these microglial cells will change their behavior and they change their morphology. You can see in the right-hand side, this pink microglia looks quite different to the left one, which has elongated processes, and really fine elongated processes and small cell body, whereas we start to have much bigger cell body and bunchy processes for these activated microglial cells, which is associated with the production of inflammatory mediators. Now, if you see these interactions in the brain between neurons and microglia, these are quite diverse, and you can see they appear everywhere in the tissue, allowing the proper monitoring of neuronal state and, for example, the incidence of infection by these cells. And I don't want to spend too much time on it, but defining these interactions remains extremely complex. And now we are looking at the level of two membranes, the microglial membrane, which is here, the green, and the neuronal membrane. And between these two membranes, there is extremely fine-tuned interaction based on the release of simple mediators like ATP from the neurons, providing clues for microglia to sense if a neuron is injured or if there is any kind of disturbance in the brain microenvironment. So when I, as a neuroimmunologist, started to work on my PhD, initially we wanted to explore how the nervous system regulates different immune organs. And a good model to do so is to use a virus which uses a neuronal pathway to spread across synaptically linked neuronal circuits. And if you inject this virus into peripheral organs, immune organs, or any kind of organs like the spleen or the bone marrow, what the virus will do, it will retrogradely go back to first the spinal cord and then to the brainstem and to other areas of the brain, allowing us to map those neurons which are synaptically linked and regulate the functioning of different organs. And while it was an anatomical study, there was a very interesting observation that over time, these viral infected neurons started to disappear from the viral infected areas. And it turned out that there are very strange little cells which surround these neurons. And we could also, by using markers that demonstrated whether it's a newly infected neuron or it was infected longer time ago, allowed us to actually identify that this is a relatively rapid response while microglial cells are recruited around these infected neurons and simply eat them up. And at that time, it was an idea that this might provide a very good opportunity to control viral infection in the brain. As these microglial cells, the red cells here around this blue-green neuron, provide seemingly a tight barrier, and you can also see the products of the degenerated neurons inside the microglial cells. Now, I had to wait 15 years for technology to develop and to be able to record this in vivo in real time. So what you see is a sleeping mouse, and the mouse received a similar viral infection. Now the virus is red, and you can see this red dot in the middle. This is a single neuron infected by the virus, and you can see these microglial cells surrounding it and forming a barrier-like structure and start to rip it into pieces. And this is quite a fast response. It goes on within a few hours, and you can see these images reconstructed here on the left side. And we have now evidence that actively these microglial cells, phagocytoses, eat these neurons, and through this, they actually control viral infection in the brain. And you will understand a little bit later why I'm telling you this story, because we have tools to remove the microglial cells from the brain. We have multiple tools to selectively tune microglial activity. And in this case, if you remove microglial cells in the brain, you can see that these yellow cells disappear in the depleted group. Viral infection runs away, and you will see very, very severe neurological symptoms in these animals compared to those which have normal functioning microglia. And at the same time, you start seeing the viral proteins appearing in the brain parenchyma. So the lack of proper brain immune function, in other words, would lead to an uncontrolled spread of an infection which is in the brain. And this is coming to be very important in a number of diseases. A very typical example is herpes viruses, which very rarely cause encephalitis, but it seems that other viruses, such as coronaviruses, might also trigger a similar immune response in the brain if they get there. And if this immune response is not properly functioning, you can have serious diseases and complications as a result. Now, before we go further, I would like also to take the opportunity to tell you a little bit about how actually immune cells like microglia or peripheral immune cells like macrophages recognize infection. Because it would be very difficult otherwise to imagine how these cells can recognize infectious organisms unless we identify some molecules which are specifically linked to these infectious organisms, but are not found in the human body, for example. And this is actually the case. So detection of pathogens is performed through so-called pumps or pathogen-associated molecular patterns. For example, viral proteins, viral DNA, viral RNA, which are not found in the body. So if an immune cell like a microglia or a macrophage would see these specific molecules, it would trigger inflammation straight away. And this would be a straight sign for the cell that there is infection, and this is why an inflammatory response needs to be initiated. At the same time, from the injured tissue, there will be another set of molecules, which we call DAMPs or damage-associated molecular patterns, which will get released, for example, from injured cells like neurons. These can be very simple molecules. And if they get into the environment, microenvironment, it is the same thing in the lung, for example. If there's a lung infection, first, there is the infectious organism itself, which would trigger inflammation. Second, the infected cell, the degenerating infected cell, would straight away release other types of molecules, and they collectively alert the immune cells, which make their surveillance activity around. And there will be an inflammatory reaction. And this inflammatory reaction is an intentional reaction of the body to coordinate the elimination of the infectious agent and also to support the restoration of normal tissue function. So let's have a look at the inflammatory signs like heat, redness, swelling, pain, and finally loss of function. This happens when, for example, a bacterial or viral infection reaches the lung. In this case, there are no microglial cells, but we have macrophages and neutrophil granulocytes, which will be the first line of defense in the lung, for example, circulating also in the blood. And these cells will notice that the infection is there. They'll start releasing different inflammatory mediators, which will alter the permeability of blood vessels, allowing immune cells and inflammatory mediators to get out to the tissue. And that initiates a coordinated response, which initially causes swelling, redness, then it will trigger the sensory nerve endings which are in the tissue, which will cause pain, and then you will see that this ödemic tissue, which is really hot, won't function properly. So, for example, you won't be able to move your finger after a cut. And this inflammatory response is very well coordinated, but first it remains local. After that, if it reaches a certain threshold, for example, in the case of a lung infection, like we see after coronavirus infection, this inflammation will need supply from peripheral immune organs, and there will be a coordinated inflammatory response which would start affecting the whole body in order to recruit immune cells and also to modify certain physiological processes to tolerate the effect of infection and to fight infection properly. And what happens is that as the permeability of blood vessels change and immune cells start to get activated, these inflammatory mediators leak out to the circulation. They will reach organs like the liver, and the liver has a specific apparatus to initiate a so-called acute phase response when there is inflammation in the circulation. This will then have an impact on coagulatory processes in the blood. This will then change circulation, and these factors then also reach the brain, and inflammatory factors reaching those open areas in the brain I mentioned, the circumventricular organs and those structures which are linked with them, like the hypothalamus, will initiate responses which we all know. If we catch the flu, we experience fever, we experience hyperalgesia, we change our normal activity, we don't really want to eat that much. These are all actions on the brain by these inflammatory mediators, and the overall aim is to have a coordinated response, allowing immune cells to function, but at the same time to limit energy expenditure concerning other processes and to fight infection properly. And then once this inflammatory response reaches the brain, a lot of other things will change, because the brain would start to initiate a defense response, kind of triggering a different level of activity, altering metabolic status, altering energy expenditure, and you will see how much it can impact on your daily life. And once the infection is over, in a few days, it returns back to normal. In the meantime, this brain response is also important to coordinate the inflammatory responses of the peripheral organs. And I don't want to spend too much time on it, but the take-home message is through neuronal and humoral routes, so through the autonomic nervous system and through the neuroendocrine system, this altered brain activity will start controlling all the inflammatory processes in the body, and it will affect how active immune cells are, with the overall aim of mounting a proper immune response and to fight infection. Now, the problem with any kind of inflammation and infection that you have to set proper limits. There are controlled forms of fire. In the case of big fire, it can be controlled, and there are uncontrolled forms of fire, and we know that uncontrolled fire can cause a lot of troubles. Now, this is the same system or situation with inflammation and with inflammatory diseases as such. And this takes us to an additional topic, which I really want to touch upon briefly, why we have so different responses to infection, and why the inflammatory conditions are so frequent in our life these days as compared to the life which others experienced like 100 or 200 years ago, and through the history of humanity. If you look back 100 years ago, we didn't have antibiotics, we didn't have proper medications, we didn't have immunization, and a lot of other things. So the majority of the people died as a result of acute or chronic infectious diseases, tuberculosis, pneumonia, diarrhea, and a lot of other conditions. And over 100 years or so, through the advances of medicine, many of these acute conditions, many of the infectious conditions disappeared from developed countries, although they didn't disappear completely, as we can see this in the case of the coronavirus epidemic. But as a matter of fact, if you look at this pie chart below, these days, heart disease, cancer, and stroke would take 2/3 of the total deaths as compared to the turning point 100 years ago. And this, among a lot of issues to consider, brings some new challenges to us, and this would have a lot of crosstalks with evolutionarily conserved mechanisms which have not been properly eliminated, and this is why they would impact on our body differently as they did hundreds or thousands years ago. And just one example to bring to you is coagulation, and you will understand very soon why I'm talking about it. For example, if you look at insects which have a really open circulation, so they don't have their blood circulating inside blood vessels, but they have a hemolymph which would filter their organs. If you want to protect an insect body against bacterial infection, for example, you need to act differently. And among many other things that you alert immune cells, you need to restrict infection, and as such, insects use coagulation to control infection. If there is infection in any peripheral organs of an insect, there will be immune cells activated, as we see it in our body, partially using similar molecular patterns, responding to similar molecular patterns of the infectious organism. But there will be coagulation, and in other words, coagulation is evolutionarily linked to infection and inflammation. Now, this causes a lot of problems in our body when we are exposed to too much fat, or we experience too much stress, and there is an increased burden of atherosclerosis and diabetes and other chronic conditions. At the same time, we live much longer, and this body will respond differently to infection. And for example, one factor which is going to be exacerbated is coagulation. And as we get older, the control over this process is less and less. So aged people would be more exposed to coagulatory problems than young people, for example, which will be, for example, a very important issue in morbidity and mortality concerning the coronavirus infection, as you will see. At the same time, if we go back to the brain, we learned that we cannot really treat brain disorders. And when we wanted to protect neurons in the brain, these trials all failed. And one of the reasons for that is that inflammation is a very common condition which is part of all brain disorders, but at the same time, our inflammatory responses are not normal in a way, because we are exposed to so much chronic inflammation. Our body works differently, and if we catch if we catch an infection, this body will respond completely differently to infection as if we were living in a natural environment. Where can we get examples that, for example, infections contribute to other diseases, and how can this work? A typical example is stroke, and the causal factor or causal role of infections in stroke, for example. There were many studies. I'm just spotting one. This is the Northern Manhattan stroke study, in which researchers looked at associations between infectious burden, and stroke incidence. And as you can see, infections increase stroke incidence, but also in seasonal flu, you see the same thing. The incidence of stroke and the severity of stroke is higher if there is preceding infection. At the same time, many patients in the hospital with a stroke will have a dampened immune response, and opportunistic pathogens can colonize the body, and a vast majority of people who would not die as a result of a stroke would die as a result of infection because the immune system is not functioning properly. So it is very important to keep a proper control over immune processes, and chronic inflammation or brain injury could interfere with this. Now, wouldn't it be easier then to eliminate inflammatory cells from the brain? Because if we think that this is due to inflammation, which are the inflammatory cells in the brain which I showed you to protect against viral infection? These are microglial cells. What happens if we take them out? We have tools to selectively eliminate microglia from the brain. What happens if we induce an experimental stroke in a mouse? You can see that as opposed to the expectation, this stroke is far more severe. So we need these inflammatory cells in the brain to function and to control even an acute injury like a stroke, but at the same time, they can also cause inflammation. So how does it work? This goes back to one of the stories we have been doing in Manchester with my good friends, Stuart Allen and Nancy Rothwell. Nancy Rothwell was actually the first one who discovered how inflammation impacts on the brain and how inflammatory processes can cause brain injury. And when I was in Manchester, first after 2005 and then from 2008, we have been doing a lot of studies. Many of them are still not finished, to define the mechanisms through this inflammatory disease's impact on the brain. This is a PET scan from patients who have chronic inflammation based on this inflammatory marker CRP, and also they have multiple risk factors for stroke. And the color things in the brain, you can see these are indicative of microglial activity. And when a patient has risk factors for stroke and chronic inflammation, these microglial cells change their activity, and they will respond completely differently if there is an infection or injury. We model this in animals as well. These are so-called corpulent rats, which are obese, atherosclerotic, and kind of resemble a human metabolic disease. You can see they weigh approximately 1 kilo. Compared to their lean counterparts. And as they age, these animals also display inflammation. They display altered microglial reactivity in their brains. So in other words, if there is a challenge, they will respond differently. For example, these animals have much more severe stroke outcome if you induce an experimental stroke in them. How does it work? If there is inflammation, you will have a much higher level of blood-brain barrier injury. So those fine structures, which I showed you at the beginning, which control the communication between the blood and the brain, will deteriorate, and then nerve cells cannot function properly. At the same time, inflammation in the systemic circulation will also change the perfusion of the brain. So if there is inflammation, for example, if there is a lung infection and there is high level of inflammation, this inflammation combined with the stroke will reduce perfusion, meaning that these brains will get much less blood supply, and the injury which develops will be much more severe. And this leads us to studies which we performed on the mechanisms to define how these preceding infections impact on brain injury and stroke outcome. And one particular study which we performed used a human pathogen it's a bacterium, Streptococcus pneumoniae, and this gives a very similar infection or infectious pattern to that of coronavirus, because this will remain in the lung. So initially, coronavirus also infects the lung, and this Streptococcus strain will not spread to other tissues in the body. It will stay in the lung, but it will induce a systemic inflammatory response. As you recall, it will connect to the liver, and then all the immune organs will have an altered activity. Immune cells are activated. They will produce a lot of inflammatory mediators, and we will have a higher level of inflammatory load in our circulation, which will reach our peripheral organs, and it will reach also the brain. What impact this higher inflammatory burden will have on different pathogenic processes which we experience. On the left-hand side, you can see an image from the aorta. When we feed the mice an atherogenic diet, you can see that if there is Streptococcus infection in the lung at the same time, there are much higher level of plaques in the aorta. And at the same time, if you look at this picture in the right-hand side, this showing brain vessels and the marker for vascular activation, and simply by causing a lung infection and generating systemic inflammation, you will activate brain vasculature, and you will cause inflammation in the brain vasculature through these actions. What happens if we induce a stroke in these animals? You can see that infarct volume, the injured area in the brain, becomes larger, and these animals also present much worse neurological symptoms. If we block inflammatory mediators, we can pull back this extra injury caused by the preceding infection. What else can we see? We can also see that platelets, and these are the cells which contribute, or cell-like elements contributing to coagulation, will aggregate also more in brain capillaries. And you might recall that I told you that coagulatory processes are evolutionarily conserved in a way, and they linked with inflammation. So preceding infection and systemic inflammation caused by infection will also alter aggregation of these platelets in brain microvessels. If we block this, we can reduce also brain injury. So if there is inflammation in the systemic circulation, this will affect all organs, it will affect the brain, and it will make brain injury more severe. Now, this was found very similar in the case of influenza virus infection, which study was performed by a good colleague of mine. As the infection propagates, if you induce acute brain injury, a stroke, it will be more and more severe as time passes. And this brings us to the coronavirus epidemic, and within this to maybe get some clues how the infection may contribute to central nervous system injury, and how this interaction between the central nervous system and coronavirus infection could contribute to overall outcome and could cause a poor outcome in many of the patients. And I want to really emphasize that there is very little data at present, and this is why I found it more important to actually talk about the basic mechanisms, how infection, inflammation, and injury in the brain interact, because what I will show you is based on the assumption that the coronavirus infection will probably trigger very similar responses in the brain. We have some evidence to that already. And this is why we can conclude that inflammation, either caused by direct infection in the brain or caused by systemic inflammation, could contribute substantially to outcome. So if you look at the present coronavirus crisis, this is not a unique event because it was preceded by other epidemics, although these were better controlled. If you recall the SARS epidemic in 2003 or the Middle East respiratory virus epidemic in 2012, these all originated from animals, transferred to a host which didn't experience this condition. And it is very important to point out that the human body recognizes coronavirus infection. So if we get seasonal infections, 1/3 of the common infections in cold weather are caused by coronaviruses, not the SARS-CoV-2, but other coronavirus strains. So we know that they are able to cause infection, they are able to initiate inflammation, but certainly every single virus would act differently, and the immune system which is mounted to fight the infection will be different. How does it work in this case? We have virus infection which primarily would affect the airways and cause infection and inflammation in the lung. As you might recall, other infectious organisms, such as Streptococcus pneumoniae I showed you, would induce a really robust inflammatory response in the systemic circulation if the lung infection reaches a certain threshold. And initially, this response, which will also reach the brain, is intended to coordinate these inflammatory responses and to fight infection. And now what you can see, we have in coronavirus infection a lot of symptoms which are associated with lung infection, like chest pain, shortness of breath, dyspnea. But we also have a lot of infections which are related to the interactions between these inflammatory mediators which are in the circulation and the brain, such as fever or anorexia or vomiting or diarrhea and other symptoms, which will already show you that this infection reaches the brain through at least the inflammatory pathways, which are quite common in any kind of conditions where the level of infection reaches a certain threshold. Now, what other things we can learn from the published materials? We also have symptoms in different organs because there are certain conditions when the level of inflammation reaches a certain level and it becomes systemic, it can affect many organs in the body, and it can, in severe cases, cause multi-organ failure. This is very similar to that seen in, for example, bacterial sepsis, when there is an excessive production of inflammatory mediators, and this high level of inflammation will have a number of changes which are very difficult to tolerate. And I would like to really call your attention on this, because resistance to infection is quite different compared to tolerance to infection. So we might resist very nicely, our immune system can restrict the spread of the virus, but we might tolerate it less. And this is what aged people experience, not just in response to infections, but in response to other challenges. There's a certain level of infection, but it's very difficult to tolerate it. It will have a stronger impact on the body. And even in young people, this excessive systemic inflammatory response, which is initiated after coronavirus infection, has a really strong impact on different organs and on different systems. What are these? For example, there will be altered coagulatory processes, so the blood will clot very easily. And this is why we can have heart attack, we can have stroke, and also several organs like the liver or the kidney can have a deficient circulation, and certainly failure of these organs can also cause additional severe symptoms, such as we have accumulation of toxins, and these toxins will have additional impact on an excessive inflammation and a really life-threatening, dangerous condition, especially if the infection at the same time is present and it pushes towards an even more expressive inflammatory condition in the body. But at the same time, it will also reach the brain, and in rare cases, we could see that encephalitis is a condition which comes to be present in coronavirus infected patients in severe cases. And if you remember what I told you about the regulation of inflammation, it is especially true in this case because this is a virus which the human body has never seen. So our responses are already atypical to some extent. At the same time, the virus will induce a really strong inflammation in the lung, and it will really have an impact on the majority of the lung surface, which means that there is a lot of inflammation there. The body cannot eliminate the infection, and this will cause a condition which is caused cytokine storm. And I don't want to spend too much time on the mechanisms, but you can imagine that those inflammatory cells which would recognize the different elements of the virus, which are not normally found in the body, which we named PAMPs, pathogen-associated molecular patterns. These inflammatory cells will release inflammatory mediators, and these inflammatory mediators will have secondary mediators. And if this reaches a certain threshold, it is very difficult to regulate this excessive inflammation, especially if it's ongoing for many, many days or even weeks. What will happen is that the immune cell populations, which are supposed to fight infection, will be more and more difficult to generate, and the balance between these populations will be shifted. For example, our lymphocytes will start dying, and many of these lymphocytes are controlling those innate inflammatory cells like macrophages or monocytes which notice infection. So the cytokine cocktail which comes out is going to be more and more difficult to restrict because we start losing the cells which can regulate these actions. And you can easily imagine that beyond a certain point, it can reach a very severe status when coagulatory processes are altered, when circulation is altered. If the body is hypoxic, it's even more and more difficult to supply those organs with fresh blood. And at the same time, this excessive inflammation will also reach the brain. And what is really remarkable in the case of coronavirus infection is the really high level of incidence of neurological symptoms in patients compared to other viruses, which indicates that either there is a very strong inflammation, which is kind of not the case actually compared to bacterial sepsis, for example, or this virus might have other effects on the brain. For example, could interact with brain cells and cause infection, which can also come from unbiased studies looking into protein-protein interactions, such as that published by Barabás, Albert László, and others, implying the nervous system as an important target of this disease, simply based on interactions between viral proteins and other proteins in the body. Now, what kind of complications can we see, which are not necessarily due to direct infection in the brain? For example, there are reports about young people with ischemic strokes or bleeds in the brain. And this is a population which don't have chronic disease, which normally don't get stroke because coagulatory processes are well regulated. The immune system is relatively fresh, so it can fight infection. The balance between procoagulant and anticoagulant factors is maintained. Still, these people start showing strokes, coagulation, clotting in the venous system, or bleeding in the brain, which is quite strange. And here we start speculating about whether, in addition to the systemic inflammatory actions, this virus might also affect, for example, brain vessels and cause direct inflammation there. And this is actually the case. looking at the virus receptor, which the virus uses to enter the body, this angiotensin convertase enzyme 2. This is expressed, for example, in the olfactory epithelium, but also expressed on the wall of blood vessels. And it is very likely that if the viral titer reaches a certain threshold, and at the same time the immune system activity is going down because of the long-term systemic inflammation and infection maybe in other organs apart from the lung, it's easy to imagine that from also these immune cells and from other sources, the virus can reach the brain. Another possibility is that the virus reaches the brain through the nerves, which is also very commonly seen in the case of herpes viruses, as I showed you those track tracing studies we performed to define those autonomic circuits which regulate immune organs. But also with the Asian influenza virus, it has been noted that the virus can spread across synaptic linear neuronal circuits. So theoretically, from the lung, because the autonomic nervous system innervates the lung to control function of lung cells and circulation and other processes, the virus can actually reach these brain areas and cause infection there. Now, it seems from single case reports that this is the case. It seems that we find the virus in the nervous system. We find it in the brain parenchyma, which is remarkable that in the case of the Middle East coronavirus epidemic and also with the SARS epidemic, this infection was present mostly in the brainstem, where the structures controlling respiratory function, breathing, blood pressure and other physiological processes are located. So it is very likely that in this case, the viral infection can be present also in the brainstem. And also, what is confirmed already, that the virus is present in the olfactory pathways. And the problem with the olfactory epithelium, that if it's infected by the virus through the olfactory nerve, the virus can very quickly spread to the brain, because there is this olfactory bulb which would connect the axons of the olfactory nerve and then switch these to other brain structures in order to have the sensation of smell. This area of the brain is a primary target of the virus, not just because of its direct connection with the olfactory epithelium, but also because there are cells expressing the virus receptor, this ASA2, in the olfactory bulb. At the same time, MRI findings confirm that a large number of patients have hyperintensities in different areas of the brain, which would indicate that either the virus infection or the infection-induced very strong inflammation is there. Just a remarkable case report to show you, this is an MRI image of an olfactory bulb soon after the loss of smell arised, and then you can see the swelling of the olfactory bulb in the patient, and there is also data that 14 days later this goes back to normal. So it is very likely that there is infection and inflammation, and the hallmark of this inflammation in this case is the edema in the olfactory bulb. So the virus reached the brain. And just an outlook for half a minute, this is a very interesting study published just the 5th of June by an American group, showing that those microglial cells, which I mentioned to you as primary elements to control viral infection in the brain, are important also to control viral infection in the olfactory bulb. So you can see this is the cribriform plate. This is the connection between the brain and the nasal cavity. This is the bone through which the nerves of the olfactory epithelial, olfactory nerves pass through. And it seems that these microglial cells are there and are activated in response to infection. And we can, based on the previous data, assume that the functionality of these microglial cells is important to control infection here. And there are also a number of other observations showing that the viral reaches not just the olfactory bulb, but also the cortical areas, which are important for sensation of smell and for other functions. And I would make the point that in combination with the excessive systemic inflammation caused by the lung infection and maybe infection in peripheral organs, local infection in the brain could lead to a deadly cocktail. You can imagine a patient with a really disturbed lung function not being able to maintain proper oxygenation in the blood. And at the same time, in the brain, the structures which control respiration and circulation may be affected. You can imagine that this patient has all chances to do poorly after reached a certain severity of infection. And at the moment, we have very little data to show how important these brain infections are in the actual morbidity and mortality of patients, but it appears that in a number of cases, we can at least have to accept the possibility of brain infection. And once the infection is there, it will surely impact on how the brain functions. And certainly, other conditions like hemorrhages and encephalitis in certain patients indicate that the brain is a really important target for COVID-19. And it will be very, very important for us to understand the processes which regulate the viral infection and those processes through which the immune system can fight infection and regulate this excessive inflammation caused by the infection. So after this very long and tiring summary of many years of research, I would like to give you a few hints, a few points which might recap on some of the mechanisms we've been discussing. First of all, probably I could show you that inflammation either inside the brain or in the systemic circulation have profound impact on the nervous system. And we know that they would contribute to either brain inflammation or systemic inflammation would contribute to a number of conditions, not just neurological diseases, but conditions like cancer through the regulation of immune cells and immune organs. And this is something we have to really consider concerning the present coronavirus crisis. At the same time, we have microglial cells, which represent the first line of immune cell or immune defense in the brain. And these are at the same time the main regulators of central inflammatory processes, which control viral infection. And I think here we can make two categories. In case we have infection in the brain, which is still unclear how frequent it is, but if it happens, these cells and the brain inflammatory microenvironment will be extremely important to control the spread of infection. And I have told you what I would like to emphasize again. Microglial function is profoundly influenced by systemic inflammation. So those aged people, those people with diabetes, those people with hypertension, which represent the primary target group for coronavirus-related morbidity and mortality, probably also suffer from the infection that severely, because if the infection is severe enough and brain infection occurs, their microglial cells and the whole system which would control inflammation is altered. And we have a number of very good analogies from animal research and also from clinical studies showing how differently an old body or a body of a diabetic person would respond to any kind of infection. At the same time, irrespective of whether the infection occurs in the brain, this excessive systemic inflammatory response due to the infection will have a number of effects from coagulation through blood flow, blood pressure, causing circulatory failure, edema, multi-organ injury, and coma in severe cases, which can be exacerbated by local infection in the brain. At the same time, these inflammatory mediators reach the brain and will cause a number of changes there. Very typical one is fever, but also it can lead to more severe neurological symptoms, contributing to mortality of the patients. And finally, the loss of senses of smell and taste, the frequency of encephalitis, and the sudden appearance of circulatory and respiratory symptoms in patients, or those symptoms where patients don't simply sense hypoxia, might indicate that those structures in the brain which contribute to the regulation of breathing, circulation, blood pressure, and other processes are directly affected by the virus infection. And finally, I just would like to show you an analogy which we use a lot these days, how we can think about brain inflammatory cells, microglia. I use this expression, the grumpy maid. If you know the stories from Sir Arthur Conan Doyle or Agatha Christie, the maid is an important element of the house. Without the maid, nothing functions. But if you wind up the maid, it can put some poison into your tea, and certainly this is a bad news for the lord of the house. And how true it is, on the left-hand side, I show you an image from a human brain. This lady was 103 years old when she died, and you can see the neurons in red, and you can see the microglial cells in green, and these microglial cells surround the neurons, and you might recall that these are moving their processes very fast and constantly survey the microenvironment. And try to imagine we counted exactly in a viral infection model that if we had infected neurons in the brain and microglial cells would get a signal to phagocytose them, these microglial cells, 5 to 10% of total cells what we have in the brain, would be able to eat our brains up within approximately 100 hours. And in this case, it didn't happen for 100 years. So these processes have to be really tightly controlled and regulated. And the last thing I would say is that what we find important in inflammatory diseases, and especially in brain diseases, is that as long as you have regulation of inflammation, things don't get very severe. And the real problem comes when the regulation is gone. And I think this is a really important thing in the case of the coronavirus disease. And we have a lot to learn because most of the data are still not available. So with this, I just want to thank the people in the lab who really give most of their life to pave the way of the studies we pursue. And also for founders, ERC, and I would like to really emphasize the Landulet program, because this allowed me to come back from the United Kingdom, and this allowed me to establish my lab here, and allowed me to write up a successful ERC proposal, showing that it was not 100% pointless to initiate this really excellent program from the academia. And I would like to thank your attention and happy to respond to any questions. Thank you.
Thank you very much. Are there any questions first? Well, there were a handful of new concepts and even names for us.
Actually, I have a question. This is Gergely Szakács.
Okay, go ahead.
I don't know if I can ask a question, you know, from being a non-mathematician.
Go ahead.
I try to keep my question very general so that it is of broad interest. First, maybe a remark, what you said, Ádám, at the end about the balance and regulation is exactly the same for cancer. So we have these two opposing mechanisms in our bodies which need to be kept in balance, otherwise disease happens. So the question is this: I just heard from a friend in Sweden that an emerging concept is that in line with what you just showed on your slide, that there are other types of coronavirus. And the concept is that there might be cross immunity. So some people who may have been infected with earlier versions of this virus may be immune to COVID-19. So do you know anything about that?
Yeah, I mean, there are a few studies about this. Unfortunately, I mean, they are not powered properly, at least to my taste. So this is kind of the same argument as vaccination against tuberculosis would change the kinetics of infection. But I think it's really important to speculate about these because, as I mentioned, and probably you all know, the common cold in 30% of the cases is caused by coronaviruses. So probably if you know coronaviruses, other strains very well, such as the different forms of flu, and maybe the types we were exposed to and how we actually deal with it could be important. At the same time, I think this would not alone explain this huge difference we see concerning the burden of infection in different countries, if you just look at European countries. So it would be, to me, very difficult to imagine that this would be the sole case for it, but it's definitely a contributor.
Any other questions?
I don't see any other questions.
So I killed completely the audience with this majority of information.
Well, at least it helps. You know, people might think that they are already virologists or immunologists, but it turns out that there is a lot more into that. So we thank you for this very informative talk. And before finishing, let me announce our next meeting will be next Monday.